One of the important programs that we're working on is related to trying to improve drug therapy. After, let's say, a woman is treated for a newly diagnosed breast cancer and has surgery and has radiation treatments and all the tumor appears to be gone, many women are given drug therapy at that point in time, not even really knowing whether there's cancer present, hidden away somewhere in the body or not. And the idea is to kill the cancer cells when they're still in very small deposits present somewhere hidden away in the body. Now let's take a look at what our options are for drug therapy. Those drugs are given intravenously and they circulate throughout the whole body, and they are toxic to our whole body. They're not selective for cancer cells generally. They kill cells that are rapidly dividing. So hair cells get killed and the hair falls out. Our immune system is rapidly dividing cells and they get killed and our immune system gets pushed way down. And so these are the limitations for our current, what we call cytotoxic cancer drugs. Now wouldn't it be wonderful if we could somehow modify those cancer drug molecules so that every time they went by a cancer cell, they would stick there and accumulate there until that cancer cell was just covered with a high concentration of the chemotherapeutic drug, and the rest of the cells would just float on by. Well, that's what our laboratory is working on, and this is really one of our main efforts at this point, is to try and generate a customized cancer cell binding molecule. And we're using a technique to accomplish this whereby we have generated what we call a library of different shaped molecules. And these are made randomly. They have different sizes and different properties. And the idea is that if we made enough of these, we would find some that would stick preferentially to the cancer cell. Now, how many of these special molecules would we need to make? How many different ones would we need to make? Well, a lot. And the power of modern molecular biologic techniques allow us to make huge numbers of different shaped molecules. And so one of our libraries would readily have more than 1 million different shaped molecules. And the idea is we take a target, like a cancer cell, and we add our entire library, 1 million different molecules onto this, and let them react, and a few are bound to stick. Then we collect those that stick and rinse off those that didn't stick, and find out what the heck it is that binds to those cancer cells. And then we can make lots and lots of that as a selective binder to cancer cells. So it sounds pretty simple, and in some respects the idea is very simple. The complexity is pulling all that off, making all that happen, and there's many, many complex steps and challenges to meet those goals. But one way is to do like I just described, take pure cancer cells in a dish, in a lab, and add our library. And we do that and we get binders, and that's one step. But it turns out that when cancer cells are removed from a person, they change. They're not like it was in the person. And so one of the projects are that we administer our library intravenously, and then it circulates throughout the whole body. And we do that just prior to surgery, so that after a few hours of circulation, we remove the cancer. And what should be in that cancer? Whatever molecule is stuck to that cancer. And so we run that back to the lab and recover those special molecules that stuck to that cancer selected. And this is the first time this has been done in the world, whereby patients have been administered these phage libraries multiple times and allow them to be recovered and find out what these are. So where is this going? Well, we want to make a system whereby we can generate a customized set of special binders to anybody's cancer. And I don't see why that can't happen. So we work through the steps one by one, and And we've made a lot of headway in working towards this process.